Abren de miércoles a domingo. Sí, porque me metí para ver.
¿Te hubiera traído la tabla?
Más si se la pasan ahí. Sí, ya les di el teléfono, están ahí mensajeándose con Billy. No, si van a vivir toda su vida con una cosa así. ¿Qué conciertos no hay hoy?
La de el museo de Peña, ¿sí?
Concierto para violín número 1 de Tchaikovsky. Sí, no, pero es de mayo.
Muchos conciertos, actividades para adolescentes y niños.
En CEU.
Actividades para adolescentes y niños.
Uno, dos, tres, probando. Uno, dos, tres.
Está el Tim Burton.
Sí, es
En la mesa.
Magia, ¿qué? Fantasía.
No,
Está
pero
lo
ese
de
es de Tim Burton.
Tim Burton.
Ah, pues también ese.
No,
Ese
ese
está
ni les
bueno,
gusta.
ese está bueno también.
Hi, everyone. Thanks for being here. For me, it's a great pleasure to introduce Carlos. Carlos studied biochemistry at the Instituto Politécnico Nacional in Mexico and obtained his PhD in biotechnology at the Max Planck Institute in Germany, where he also held postdoctoral positions. He later joined the Danish biotech company Biosyntia, co-developing the world's first process to produce B vitamins from renewable sources instead of fossil materials. Since 2023, he leads the Computational Protein Engineering Group and the Novo Nordisk Foundation Center for Biosustainability at the Technical University of Denmark. His research focuses on improving existing and designing novel proteins using modern tools like AI to create more sustainable biosolutions for society while protecting the environment and human health. Well, thanks, Carlos, for being here, and the stage is yours.
Thank you very much, Sergio, for allowing me to come here and present my work. I just started my group 2 years ago, so I don't have that much results, right, as also Sergio. Recently, he joined UNAM, but I would like to present you some of the work that I've been doing in the last, 15 years in Germany, but also in Denmark. And it's the first time actually that I come to UNAM to give a talk, so this is a privilege for me. Also that my family is in the audience, and it's a bit, exciting. So I'll like to, before I go into the topic, I'd like to present very general what already Sergio said, which is more or less my academic career. What I would like to highlight is that I work in the industry, and most of the people change from academia to the industry. I decided to go back because there were still many challenges to be solved, and leading a team has been always a dream for me, learning from the young people and how we can solve problems, because when you're in the industry, it's very much driven towards the application, and you don't have time to go and to study things in more detail. And moreover, I have been asked, what is more stressing, Carlos, working in academia or industry? The industry is very stressing because you have to give results every 6 to 8 weeks. And if you don't satisfy the investors or the board of directors, then you can just, become irrelevant. So you need to be very creative. And so for those of you who don't know where is Denmark exactly, here's Europe, and then there is Germany, and then in the north of Germany there is this peninsula, which is in the west, and then in the east, this is the island, Sjælland it's called. And there are a lot of campuses from DTU. We are located in Lyngby, which is in the north. And it's a beautiful area, green lakes, and very close to the sea. Now, if you look at this university, worldwide, it has been ranked among the 20th to the 200 best, depending on different rankings, which take into account papers, patents, prizes, but sustainability is one that is very strong for Denmark, and that has been, especially DTU, very strong on that area. Moreover, if you look also into biotechnology, in the Shanghai ranking, 3 years ago, DTU was on number 3rd, which is below Harvard, MIT, and Stanford. So that's actually quite good to see that Denmark has been investing in biotechnology for many years. And this is one of the reasons why I also decided to move from Germany to Denmark, because I realized there is an ecosystem. Industry talk to academia, academia talk to hospitals, and basically it's a great place because science is not longer, just from public institutions. Industry makes a lot of research, and this moves along many different areas and developments. Those rankings are based on different things, what kind of impact of the research, world-class faculty, output, etc. Now, if you come to DTU, and you're welcome to visit me, you will go to the campus. Now it's October, so you'll see these beautiful leaves. I really love autumn there. And there in the back, there is the so-called Gucci building. It's a very fancy place because the foundation on all this gave a lot of money to build this. And they wanted to have like a center where, we can develop biotechnologies to produce the chemicals, materials, everything, medicines for the future. And so if you come inside, before we go inside this, there is different areas. We produce natural products using microbes, but also we can eat microbes. So there is, for example, hamburgers that you can eat just based on mycelium, and they are more environmentally friendly. So they have 98% less water and CO2 production and so on. We also make chemicals, and so we have been developing a lot of technologies in this center for the last 15 years. If you go inside the center, you will see this very nice E. coli glass sculpture which was designed by a famous artist, and a lot of different celebrations here. What I love is also the glass walls. Here you can see the DNA helix that extends to the 5th floor, so it's like 20 meters. Here used to be my office, my colleagues working. But what is very impressive is that you go into the main entrance. There is this wall of fame. There has been almost 40 companies co-founded in the last 40 years. So if you have a brilliant idea, you just need to apply to get funding. And if you are successful, you will get some coaching. Then you will explore some of the, entrepreneurial ideas. So Biosynthia, where I worked many years, was co-founded in 2012 by a master's student and his supervisor. And that's still alive, employing many people along those years. This is a picture from February this year. I'm here with my colleagues from all over the world, and we were celebrating because we got some funding. Donato knows Max. He works in this corporation, DTU Tec de Monterrey, and we have been having a very successful team. I don't have time, unfortunately, to explain all the projects or what we do. I'll maybe mention a few of those that you get an idea. Maybe in the future, I come back and we can tell you more about those projects. My lab is divided in so-called experiments and computers, where we leverage a lot of different disciplines to study proteins, to engineer them, and to develop new processes that are more sustainable in different ways. We make data, we test hypotheses, we develop biosensors, we produce medicines, foodstuffs. We also have pipelines to analyze data from nanopore, for instance, but also develop software. I'll mention an example about it. We also model proteins using molecular dynamic simulations and stuff like that. Here's just a glance of what we do, and this is based on a publication I will mention a bit later. How can we engineer proteins to reach the optimum? We do CO2 fixation, we do alkaloid production, and so on. We have cell factories. In this case, it's a yeast that can produce medicines. We have just developed a process to produce a molecule that can be used to treat Alzheimer's disease. It's FDA approved, so we have come with a new method to do this in yeast, produce, purify, and use instead of harvesting them from plants because the chemical synthesis is very tough. We also have cascade reactions with different enzymes with a cooperation with the industry. We do fragrance synthesis, and that has been just starting now for the next 3 years. This is from the wet lab from the top, from the dry lab. And what I'm most excited about is how can we actually predict dynamics? So proteins dance. They dance according to the different reaction they are carrying up on. And if we could predict from sequence activity, that's the holy grail. This is where Microsoft is working right now, DeepMind, Cradle, a lot of companies, big ones in IT. So what we have good in our hands is how can we generate quickly data and actually formulate hypotheses that we can test and so on. I will mention a little bit of this. This is a software that one of my PhD students has been developing. And also new methods we can predict, for example, the effect of a given drug from the FDA on your GPCR proteins, where more than half of all drugs target GPCRs. This was published recently by one of my AI experts. Now, I can only show you, for example, this software that Jonathan has been done as part of his PhD. When you go to the lab, you usually have a design of a gene or a cell, you build it, you test it, and then you learn. And this process is iterated until you can find something that is better. And so he wanted to support the learn and the design steps. How? By using different tools. So there are four modules. This module is called structure-based design. This is protein pin, and this is one of the tools that David Baker developed in the past, a few years ago. And usually you can design proteins, and Sergio has been doing this very nicely, that are more stable for processes in the industry that require temperature, more soluble, better expressed. And now people can just go, give the sequence, and do with a click. If you don't know how to code, like me, because I lead a group that is full of computation, but I didn't have the time or the interest in learning, you cannot always do everything right. But now with this, I can do it. And so basically, it's a user-friendly app that anybody can use. It's open source. You can actually access it. With the link is proteusai.bio. And there is another module based on, for example, Facebook has developed so-called protein knowledge models and many other academic labs as well that can predict what would be the best substitution for a given protein amino acid to improve a given property. And if you don't have data of your protein, you can use this to more or less understand the landscape, that you can choose where to mutate and what to mutate for. Once you have generated, for example, sequences or different data points, you can start doing so-called directed evolution guided by machine learning. And then you can iterate a given protein and so on. And then you can also do protein discovery using the same language models that I mentioned.
Yeah, just to understand this part, based on the diagram that you presented before, it seems to me that you are assuming that the user will provide you with the previous data that will give you a hint to the software, which mutations work the best for improving a particular activity.
Yes.
That's the philosophy behind this.
Yes.
Data-driven.
So it's both. It's both. So you don't have data, you can just go and look at the zero-shot predictions. And we have been validating this. So they're very good at guessing what not to mutate and what to mutate for. Before it was just multiple sequence alignment and you more or less guess. These are really good nowadays.
So
companies and they told Carlos, we're using your software and we're using this, and we have actually found mutations that we can improve enzymes that are bottlenecks in metabolic pathways. And once you have generated data, then you can start improving this with this other module. So this is just developing. I mean, this has been 1 year project, and then the idea is that we want to validate it also with experimental data. But yes, maybe going a bit back also for the people who are not very much aware of that, I wanted just to show also, I mean, as is in the title, well, the Novo protein design is an area that is being now recognized last year by the Nobel Prize to David Baker. Nowadays, you can just take the computer and design proteins basically. And it's easier to design something that can bind than something that can catalyze some chemical reaction. For those who are not very, I mean, most of the audience here know that proteins have 20 amino acids that they fold. DeepMind got also the Nobel Prize last year to predict protein structure. And all these methods have been used to train protein language AI models to predict protein function. I would like to highlight maybe an example from one of my colleagues at BTU who developed proteins to neutralize snake venoms, because this is a serious problem worldwide. I don't know if you knew, but about 100,000 people die every year of snakes bites, and more than almost 3 million will just have consequences, for example, some amputations and serious discapability issues. Previously, people were collecting snake venoms, they were immunizing horses, collecting the blood, extracting the plasma, purifying the antibodies, and then formulating that. But they were not really specific. They can create reactions that was expensive. And often people will also die if they will have the right antivenom to treat the patient. And so Susanna Batz, I don't know, maybe you know her. She did the PhD with David Baker, and they collaborated 2 years ago with Tim. I work a lot with Tim at DTU, and he's an expert in snakes and all these venoms. So they basically identify what are the toxins that produce, for example, a cobra. These toxins break cells, and basically they paralyze, and you cannot move. You die instantly. There are like small ones and big ones having different effects on the cell, and then basically using this RF diffusion to diffuse, for example, the gray protein, make a protein, a computer that can bind this toxin and neutralize it. And so they did that. They tested in vitro and also in vivo. They took some mice, they injected the proteins designed a computer before and after being eaten by a cobra. And those that didn't have the protein died, whereas the other ones survived. And this is a breakthrough. I mean, it it's not a very expensive technology. It works very well. And they want to extend this to many of the different, more than 2,000 different types of toxins from snakes. They are working on different ones here, but the idea is that they want to do binders that can bind simultaneously to many of different toxins. And this is just one nice example of, the power of protein design. Similarly, for targeting cancer or the diseases, they can be used. This was mentioned last year by David Baker in his Nobel lecture in Stockholm. And that was also the great work by Susanna. I don't know her. I think she moved recently to Spain. She didn't want to come to Denmark. It's quite cold and dark. I can understand that. But we invited Kira Sumira, who did the PhD also with David Baker. She came to the DTU Biosustain and she gave a presentation how you can design also enzymes that can degrade plastic. And I'm fascinated by enzymes. We were mainly with enzymes in our institute. What she did was, again, applying RF diffusion based on a fuel and design, and then with different AI tools. Basically, she got into designing not only functional enzymes, but also enzymes having different scaffolds that could have an activity going from very little, 180 to 1,000 times more activity depending on the scaffold, catalytic turnover. And so now we're in a place where we can design enzymes and proteins for many different applications. And I don't know if you are very aware of the power of enzymes, but actually they are used everywhere to control pests, to process food, feed, in health, not only for diagnostics, but also for treating different diseases, making pharmaceuticals, chemicals, and also in detergents. They are spread everywhere. And I like to mention that Denmark exports around $1.4 billion. That was just 2 years ago, based on enzymes. So the Denmark depends on these products a lot. And the demand for more sustainable processes, it has been shown, they consume less energy, they can be more efficient in terms of CO2 production and so on. It's growing with a rate of 8% per year, and they generate a lot of direct and indirect jobs. So enzymes are really important, and that's what I would like now to continue, maybe to introduce you to the world of enzymes and what they can do. I'll skip maybe a few of these slides. Just for those of you who don't work with cells, I will talk today about enzyme engineering and metabolic engineering. When you do metabolic engineering, you have E. coli, which is just not a small microbe. It's full of proteins, right? They are very crowded environments, and you want this bacterium to stop growing, well, grow, but then produce your product of interest. That's quite hard, given the complexity of these so-called machines. But we have been doing good. I'll skip this because it's a bit more basic stuff. Importantly, a lot of the enzymes that we want to engineer in metabolic pathways, as you know, they bind substrate, they catalyze a reaction, they form the transition state, they release the product, and this is repeated. But not all the enzymes are equal. If you look at the 1,000 enzymes, for example, from Brenta database, the median catalytic turnover, so the efficiency of converting something to a product, is here Rubisco. We are taught in the textbooks that Rubisco is quite slow, but actually this is in the median side. I mean, there are a lot of different enzymes in the left side, from natural product biosynthesis, secondary metabolism, that actually are worse. And a lot of those enzymes are important to produce a lot of chemicals and materials. So we're really facing here a dilemma. We want to have biotechnology because it has so many benefits, but it's quite hard. So here is where we can use all these new methods, not only computational design, but also direct evolution that I will just introduce in a minute. When you talk about protein engineering, there is basically a collection of methods, depending on how big is your library that you want to test variants and how much you know about the system. I worked many years in direct evolution. It was awarded a Nobel Prize 2018 to Frances Arnold because she developed that in the 90s. Well, now with David Baker also getting the prize, we have been now having better tools to engineer proteins and enzymes. And for those of you who have not so much experience with directed evolution, you basically have a gene, you introduce mutations, you have a collection of variants, you screen them, express them first or in E. coli, you test them, and then the best one you can use to repeat the process. So basically you are iterating. I'll just change, I think this is getting out of, yeah, now it's getting better. And this is mimicking basically Darwinian evolution. It works very well in the test tube. This is just a quick timeline. It has been long there since the 60s. Many people got the Nobel Prize. Here is Frances when she was younger. This is Manfred Retz. I did a postdoc with him, a German scientist. He was the first one in Germany introducing these methods. And when you are engineering proteins, you know proteins have 20 amino acids. So if you're mutating 5 residues, that's already a lot of combinations, right? 3.2 million. You might just choose maybe half or even 2 residues to mutate to. This is more doable, but this is not too many. When you want to develop an enzyme commercially, you need to introduce maybe 10, 20, 30 mutations. And that means that as you increase the number of residues, you will have complexity. So how you navigate this complexity? What Manfred Retz did is, okay, if you want to go here and improve a function, given the combinatorial space, people were doing stepwise mutagenesis. How can you jump here or make it in groups? So Manfred proposed, okay, you just have to decrease the complexity. If you have an active site or something in the protein, divide it in groups, and then you can start exploring those groups in iterations. And he was inspired, according to him, by Charles Darwin, who said many years ago that, you know, the evolution of species will follow these kind of patterns. It worked very well, and you can improve many properties, as I will show in a minute. People struggle sometimes to express proteins that they are soluble. Predicting activity is very difficult still. We're working on this. We would like to improve that. Making enantiomers or new reactions or specificity is a bit easier because you need to mutate the active site of the enzyme. That works well. Stability is easier also. But often there is a problem. You need to improve many properties. And if you target one property, you might have a trade-off so that the other property goes down. And this is a challenge that not many people have solved, especially when you have 3, 4, 5 properties, also like in the snake venoms examples. So what we're working on in my group is how can we actually address these trade-offs? Sometimes you also have epistatic effects where you combine 2 mutations and you can obtain something better or something worse. And this is because they are located close to each other or they might be part of an allosteric network. So with the method from Silvia Osuna, we're trying to study allosteric networks and try to identify those. This is still very complex, but we are trying to work there. And as you improve a process, like anything in life, you can become very productive at the beginning, but sometimes you have diminishing returns. So you invest a lot of time and money, but then you don't get what you want. So these are the challenges mainly that we need to address for optimizing proteins. And maybe just to wrap up a little bit of this introduction. So a little bit of what we do in our group is, okay, use multiple sequence alignment, study protein evolution. We also study coevolution using POTS models. They have been very successful. We will publish soon some new method. We can do that. We don't work with ancestral proteins, but you can also reconstruct them and study, for example, evolution. And there are language models that have been trained also on these sequences. They can generate usually thermostable, broad activity enzymes. And this is also a fascinating area. We don't use that much often, but we are trying to use a lot of AI. And I love this figure because it shows that AI is just a broad term for different areas like machine learning, neural networks, and deep learning. We do have AlphaFold, right? I mean, this is a breakthrough that gave the Nobel Prize to DeepMind. We have 100,000 protein structures. So it was relatively easy to train those neural networks. But when we want to do, for example, propagation of KCAT activity, we don't have the data. So we're also trying to generate data, to move on how we can address these issues. What we also do a lot is using MD simulations. So how can we study bond vibrations, side chain rotations, backbone fluctuations, and so on, depending on the time scale of a given computer model that you have there, you can study your protein. And sometimes when there is a substrate that it comes into the enzyme active site, you need to understand if there are interactions, there are domains moving and so on. Because if you don't really take into account those, then you cannot really understand and therefore predict or engineer protein function. This is an example that I wanted to mention from Silvia Osuna. So what they do is MD simulations. They study the conformations that go from an inactive to an active protein. Or what you see here in this plot is many different conformations that an enzyme can do. And they have been studying what happens when you introduce mutations so that you can actually introduce mutations and promote the transition from an inactive to an active enzyme. And then you can also study the allosteric networks. And this has been powerful. They have been improving enzyme activity by many, many fold. And this is also what we want to do and use in our group. Maybe just a little bit of a commercial here. We also got a project where we could study the MD simulations, using MD simulations to study the dynamics of an enzyme that is called formate dehydrogenase. It's a dimer. It uses a cofactor, and usually it oxidizes formate to CO2 But what we wanted to do is to reverse it from CO2 reduction to formate so that we can fix CO2, the greenhouse gas. And what you see here is basically a simulation that has 100,000 atoms with waters. We were able to run this in the supercomputer of Denmark. And we are still analyzing data and trying to make sense of all this. It was a success. This computer was introduced last year. This you can see here, it's like 1500 H100 EPUs. I mean, it's a lot of power. So now Denmark wants that not only academic groups, but also the industry access to this supercomputer. And this is a picture of last year. This is the CEO of NVIDIA, Jensen Huang, the King of Denmark, and the lady who oversees this company and the supercomputer. And this has been super nice. Hopefully, maybe in 2 years, we'll be able to publish about that. This is more or less just an introduction of the basics of protein engineering, direct evolution, and so on. Let me just look at the time now because I wanted to give you some examples. So yeah, 15 minutes. So the project, so this is what I have been working on. Now I'd like to maybe quickly show you in the next minutes what I did here in Germany with Manfred Red. How can we engineer a P450? How can we then engineer a formating hydrogenase and maybe a little bit of the pathways? I can very quickly introduce you. P450s are super important for biosynthesis of everything, fatty acids, terpenes, alkaloids. So they are crucial enzymes that use a cofactor redox power to oxidize a given substrate, and they will do it in the heme domain. And there are enzymes that have many domains or just one fusing. They also degrade drugs in humans and in animals, and they also break down pollutants. So they're super important enzymes. And in our case, we use steroids because these molecules are as important after antibiotics to treat a lot of different diseases, anti-tumor, cancer, microbial, and so on. Even for COVID therapy, they were used. And when I arrived to the Reds lab many years ago, I mean, this is from 2011, they have published in Nature Chemistry that they could oxidize very selectively a steroid to give 2 different products, oxidation position 2 beta or 15 beta, 50-50 more or less. And that was a breakthrough because people couldn't actually do that before. And what they did, basically, this is the active side of the enzyme. You can see the fatty acid substrate, the heme, and there are a lot of residues that you can choose from mutagenesis. So a lady chose 3 in red, 2 in blue, and 2 in green, and started doing libraries as Reds proposed. Okay, you do first state A or B and C, and then you get mutants, and then the best mutant, you do direct evolution. You, for example, visit the site B, and so on, you accumulate mutations. And what they did basically is from 50% selectivity for either product, and then going conversion, the y-axis, they introduced 3 mutations that improve the enzyme here like that, and then here 4 mutations in 2 steps. And they had to screen almost 10,000 colonies. Of course, they have robots, and they were able to achieve this in a relatively short time. But then I was told by Manfred, Carlos, can you do it better? And can you explain me why Francis Arnold always uses this method, which is just stepwise mutagenesis, rather than introducing 3 at the same time or 2? What is better, introducing few or just 1? And so I said, well, let's try to figure out. So why is that important? Because in evolution, there is a theory that says that if you want to evolve a protein, in this case, analogy is a word, to, for example, gene, you do stepwise mutations. Otherwise, the proteins won't be functional. So you cannot go through the black arrows, only through the blue ones. And people believe also that protein evolution occurs like this. We don't know that. But if you want to study that, for example, you have 4 mutations or 7 or 6, sorry, you just create all the different intermediates. Sounds like a, easy job, but it's a lot of, it's quite consuming and kind of a bit boring. If you just need to do this for 1 year, right? Express proteins, purify, and characterize. However, you can gain very interesting insights. So we did that, and we published this 10 years later. It was a lot of work, but also, we have to study the evolution in the computer. And so we created all the intermediates. You can see going from this wild type or here to 3 isoleucines, and then trying to study why here it's important to have this hydroxy group, which, by the way, amplifies the potency of these steroids. So it's quite important in the industry to hydroxylate molecules, any molecule. And so we created a single mutants, doubles, triples, and we saw that one of the single mutants was already very good in selectivity, like huge. So there is some positions that are critical for this parameter. whereas there are other positions that are very important for activity. So different parameters have different preferences. If you combine them, then you get both very good. It's kind of obvious, but it was not when we did it. At that time, we realized that you can also make use of so-called mutability landscapes, which is nothing than mutating all your protein to the 20 amino acids in different positions. And then I did that. I created these mutability landscapes for my enzyme with 5 positions to all 20 amino acids. You can see the darker it is, the better for activity in black and for selectivity in red. And we realized that this position was key for selectivity at that position, whereas for activity, just a few. So if you don't get the key activity changes, you probably miss them. Whereas here, it was a bit easier. We did that also for position 15 beta, where we saw, okay, these 2 positions are more important, 28, 82. But we also saw there was a new position, 16, to position 82. We saw, oh, okay, this is new. And actually, we wanted to engineer the new enzymes for that position, and we were able to publish that in 2018. Why position 16? Because they can be used to make glucocorticoids, which are important for cardiovascular, metabolic, immunogenic diseases, and so on. So we showed that we could start with an enzyme that didn't have activity in position 16 alpha or beta. This is different stereochemistry, diastereochemistry. And we were able to design libraries that were different. There were 5 sites, so 10 residues compressed to 2, either green or blue. We mutated to different, we had some data, all are published. And basically what we did was screening. We found 4 mutations that were already giving great conversion, activity, and selectivity. And then we do again evolution, and we were able to get very far. We did MD simulations. We were able to explain why the molecule is going close to the oxidation compound, but we couldn't explain some of the other residues. We mutated them, and we were able to find better conversion values based on the mutability landscapes. So that was very nice. For the other case, it was easy at the beginning. Then we have this trade-off of conversion. We did simulations. We also saw, okay, this makes sense. It doesn't make sense, different interactions. We realized also that a new mutation was interacting with some of the residues of the protein, a half a helix at the top. And so there were hydrogen bonds, and we hypothesized that maybe this is not allowing the substrate to get inside. We mutated those, and we were able to improve the function of the protein, taking into account the 2 parameters, which is very difficult. Still, people are trying to figure out how can you improve many properties. And so that was hard, but it was doable. Now you know what is evolution, why people use these enzymes. Maybe just in a few minutes that I have here for MATE is so important. There is now this C1 movement where you can use formic acid to produce fuels, feed for animals, chemicals. And actually, you can generate this using renewable energy or even fossil fuels. People say, well, if you use formate, you can store, let's say, energy in form of chemistry. It can be a new dawn, for society, so that we don't depend too much on fossil fuels. And in nature, there are these enzymes, formative hydrogenases, that actually can, as I said before, interconvert formate to CO2. There are 2 different types. We use the type 1, they are easier to work with. Those other enzymes are really complicated. They have ions over cluster, they are membranes, they are big. So with this system, probably you cannot make a commercial process viable. So what we did, we took an E. coli strain that is oxotrophic for NADPH, and then we mutated the active site residues, and we were able to find mutations that were able to improve change the cofactor specificity from NAD to NADP, and that's very important for biosynthesis in cells. They need NADPH. We were able to improve the turnover, and so to make the story short, I'll just skip this now. Is we mutated the enzyme active site. The library was about 500,000 variants, but with selection. So if you can make the strain dependent on the enzyme activity, we were able to screen 4 million variants in one experiment. Here we can see we need to introduce different diversity. Each color is a DNA base, and then you mutate. Before you do evolution or screening, you need to make sure that you introduce diversity. And so this is theoretical ratios, this is experimental ratios. And we saw that when growing our bacteria with formate, they were able to survive. So we screened in one experiment 500,000 variants, and we were able to measure the doubling times, and those were basically different variants. As you can see here, from the wild type, there were changes at all these positions that we mutated using different tools. And that was very successful. We also determined the activity, the binding affinity, the catalytic efficiency, and the cofactor preference. And so this variant B9 was, and B14 was 500 better. That not only the starting point, but also what it was published at that time. And that was actually Liliana, a Mexican lady who was financed by CONACYT, doing her PhD with Aaron in Germany. And I was advising her in a kind of side project. Then we did MD simulations, and we were able to explain why these mutations were needed. For example, introducing hydrogen bonds or salt bridges to stabilize the interactions with a cofactor, or cation P interactions, and so on. So that's the nice thing, that you can combine, experiment not only in vitro, in vivo, but also in silico. And this is why it was a very nice project. But I don't have the time to go into much detail, and maybe in the last few minutes, just showing you a little bit of what I did at Biosynthia very quickly. At Biosynthia, the company was founded with the idea, why not to replace fossil fuels? We are tired of those. They are bad for the earth. Yet, a lot of the chemicals we use come from fossil fuels. All what you see here, everything in this room has been done with chemistry, basically. Even vitamins, people are not aware of that. But yes, you can use fossil fuels, you separate some chemicals, you do synthesis in China, and you can pay maybe for biotin, 1 kilo is around 1,000 euros. And we do it better. So Biosynthia wanted to develop bioprocesses that will be not dependent on fossil fuels, but let's say more feedstocks like sugars or something else, waste streams that you can actually use fermenters, grow microbes, and produce your bio-based products. In 2020, we published in the media, we were able to produce biotin using fermentation technology and purify it 98% purity. And that was awesome. We have the upstream, downstream, marketing, sales, people working together. When I was there 2016, we were 5 people. Now we're 25 or so. And how do we actually came here? Well, let's go maybe to the science, and then I can tell you a bit more of how you can get into this, commercialization. If you look into the fatty acid elongation cycle in E. coli, you make glycolysis, you end up with acetyl-CoA, and then you enter fatty acid biosynthesis. So you have here malonyl-CoA. There is a biotin enzyme that introduces a methyl group. It goes to the condensation, reduction, dehydration, by different fatty acid enzymes. So you get from 3 to 5 carbon, and you repeat this to get 7 carbon. And then you have another biotin enzyme that cleaves the methyl group. And then you have 4 enzymes that will transform this fatty acid bound to ACP to basically the precursor of biotin with this thiophenyl ring. And then you have the sulfur insertion. This reaction is the most difficult that chemists have done kind of in chemistry. And in biology, it's quite hard to do. It is done by an enzyme called biotin synthase. It has iron sulfur clusters, ethylene methionine, and this is a hard chemistry. First of all, you need NADPH, you see redox power. Those electrons will make this radical, reduce the iron sulfur cluster. You generate that radical, so SAM is converted to 5-deoxyadenosine, and this radical will allow the transfer from the sulfur, one of those cluster, to the substrate, not only once, but twice. And then you generate an inhibitor, methionine and 5-deoxyadenosine will inhibit the enzyme. And then you end up with biotin. This is what our microbes in the gut do, or many other microbes in nature. So that's quite hard to do. And when you overexpress biotin to make more biotin in cells, let's say in E. coli, you kill the cells. They don't like to do this. So we figured out, okay, how can we actually, if we generate diversity in the tube and we induce the cell to die, maybe there will be mutants that will survive because they have some kind of mechanism to cope with this toxicity, we hypothesize. And what's super interesting to see that there were some mutations that appear after a few days when there was high expression of this enzyme that is super toxic. We studied that this enzyme, it was a protein that got a transition factor that got the mutations. And that transition factor will bind to DNA to turn on the expression of many genes from different operons that called ISC and SOF. So the cells have developed mechanisms to sense stress, to sense how much iron is present. And depending on the, let's say, availability of iron and other stresses, they will turn on either of these operons. And we were able to insert one mutation in the transition factor that will be always turning on the 2 operons. That was patented as one of the first patents of the company, or the second one, and also published in 2020. And this was just, when you introduce this mutation, you will produce more iron sulfur clusters. the enzyme will be better expressed, the cells will survive, and the titer will improve by twofold, by 100% in the fermenter. And we did this many years. We created many mutants. We engineered other transition factors. We were able to improve the process to very decent level where we could actually commercialize the product to put it in cosmetics. I know it's a very short story, maybe a bit superficial. I don't have the time, but if you're interested, we can talk more. This is a picture from when I was at Biosynthia, myself here, and then a big team from all over the world. So Danes are always attracting a lot of international talent. And yeah, we have people from marketing, sales, and so on. This is when we got the Series B funding. And yeah, so we were all very happy. And so with this, I would like to end up my presentation. Maybe just please take some messages home. Biotechnology is very important. Enzymes, protein design is moving very fast. Now you have a lot of IT companies just doing a lot of biotech. Do we compete? Do we collaborate? This is open for discussion. Developing methods, not only for getting better enzymes, but also cell factories is still very important. There is this movement in Europe, that the green transition, Denmark, it's a lot of politics. People don't want to depend on fossil fuels. Are you going to depend on sugars? Yes. You might also take waste streams. You can also go to C1 metabolism, right? Okay, formate, as I explained, methanol, formaldehyde. There are a lot of options. People are working very hard to get those so that we can be more independent in what we produce and need. This concept of mutability landscapes allow for ester hydroxylation. Bayer contacted us later. They bought our libraries for 50,000 euros. Many companies are patenting all this because if you can introduce not only a, let's say, hydroxyl group, a methyl group into some drug, you will amplify the effect. So this is very, very important in the industry. You can also use genetic selection, actually also Sergio does, to quickly engineer, proteins, enzymes. You can optimize many parameters. This is amazing what you can do with very few resources. This is very important. We're also trying to develop more of those methods. And so metabolic engineering is also some key technology that will allow us to produce a lot of different, not only chemicals, materials, and so on. I'd like to thank you for your attention. Also my former collaborators when I was doing the postdoc in Germany, in Marburg, but also in Spain. I have collaborators in Bristol, in the UK. Without all these people, there would be no really good publications. We spent many years doing computations, kinetics, and so on. With the Formate story, it was Liliana, funded by Mexico. That was also a Mexican publication. We also went to work with Tobias Erb, an expert in CO2 fixation, and other people. Now I have other collaborators working with Tim, trying to introduce these protein design aspects, collaborating with people in Germany, in Denmark, in the UK. Also, we have collaborators with Harvard. I didn't mention, we also can do biosensors and so on. And let me just see the time. Yeah, 12 minutes. Should I show the video? It's like 4 minutes. Yeah, do you have time? So I also prepare, I would like to show you a video also about the center that we have been working on. It has been funded 15 years by the Nordisk Foundation, and the money runs out by the end of this year, but we will get a new funding, which is called Bright. And so they are doing a lot of marketing campaign, and they made a video so that you can have an idea of what the place has been doing the last years and also how it looks like. And yeah, so this is the last one. And then we can finish the presentation. And I will turn here the volume. This is Pablo Cruz. He's a Mexican also.
From its initiation, the Novo Nordisk Foundation Center for Biosustainability has worked towards creating more sustainable solutions to benefit humans and the planet. Here, everyone from scientists to the staff that supports them are unified by a common goal: to shape a better tomorrow by looking at the world through a new scientific lens. From day one, we have worked to harness the power of microbial biotechnology. Using synthetic biology, metabolic engineering, and data science, we have created new solutions to the problems facing our planet. At the center, we have pioneered the development of microbial cell factories, and today we are considered a world leader in this field. Using advanced computer-aided design, we can now precisely guide both strain engineering and the natural evolution of microbes. This development has revolutionized the field. Previously, it would have taken a decade to create commercially viable production strains. Now we are now, this story needs a beginning. Ours began 15 years ago. At first, there were a few of us, a select team of specialists and idealists. But since then, we have grown substantially. Today, we employ more than 300 people, and over the years, we have fostered the next generation of experts, leaders, and entrepreneurs. With a unique end-in-mind approach and a strong entrepreneurial spirit, we have generated an impressive number of spin-outs and made a remarkable impact on the Danish labor market. We are in fact one of the few places where technology transfer and translational research in industrial biotechnology delivers impact. At the center, we have developed methods and technologies for large-scale production of natural products, microbial foods, and sustainable chemicals. For instance, by delivering alternatives to dyeing clothes, new methods to keep our crops free of pests, and solutions to protect our watercourses from PFAS. With these developments, future consumers can leave greener footprints. Today, we pride ourselves in being one of the highest praised and recognized institutions within biotech research. But there is so much more we can do and explore. It is time for a change. And with a generous donation, 134.1 million euros from the Novo Nordisk Foundation, we can now make an even greater impact. Soon, we will see the dawn of BRIGHT, the new biotechnology research institute for the green transition. We will build on past competencies and experiences and expand towards new ones. At BRIGHT, we will investigate new ways based on C1 to produce sustainable chemicals and materials. We will create microbial foods that are flavorful, more sustainable, and ready for market. And we will engineer microbes for novel, effective solutions for net zero agriculture. At BRIGHT, we will revolutionize technology for biosolutions. For the future of our planet, we will develop solutions that catalyze the green transition for impact at scale. With BRIGHT, the future is indeed bright.
I mean, we look at a lot of people depend on the Nordisk Foundation. They have a lot of money for for basic and applied science, and that's based on the insulin, but also Ozempic. And I don't know what we would do without a foundation. It has been really, beneficial. Thanks for your attention, and looking forward to any questions.
Hi.
Yes.
So I'm wondering, from the many years, where to start? I mean, the question is related to which cases are you aware of where computer science has really helped experimentalists to discover something that they couldn't by mutagenesis. So, for example, Frances Arnold, as you mentioned, she showed that she could develop any protein that she wanted for any particular activity. She actually created a company with that technology, or I think she has 2 companies already.
Yes, right.
She's not using AI. So the question, is, where are the areas of opportunity for computer simulations to assist researchers in areas where they haven't so far succeed in developing solutions for applications, no?
Yeah, I love your question. Thank you very much. I mean, when I was doing this project many years ago, everybody, like Frances Arnold, everybody that does evolution of a protein, they would usually do MD simulations to try to explain the mechanism, right? And so basically, you can always look retrospectively, also in the matter of X-ray crystallography, and you can kind of, oh, it looks like this. I think almost no one, a few have used this method, MD simulation, computational method. We're not talking now about AI, just to guide, evolution. I think at least I saw, when I saw this, for me, I mean, it was very simple case. Why then a new mutation is getting here, new interactions? You wouldn't be able to see that without computation, right? And for me, it was like, oh yeah, my hypothesis, what happens when you change that? We improve this, it's nice. And nowadays we are proposing, people are working, okay, can you do MD simulations to guide protein design? And we are like, well, yeah, I mean, we know that actually, enzymes need to move, they need to have different conformations. And people are trying now to do this, but few have really shown that you can use it prospectively to do it engineer forward, right? Right now, I don't think anybody has shown convincingly. Maybe Silvia Osuna, she has used this method. She does MD simulations. She studies how well there is these atoms correlated, looking at the different variants. And then she proposed to use the SPN method. And they have shown that this year, the improvement of 6,000-fold of an activity of an enzyme that doesn't have this activity by introducing 6 mutations in one go, 6 mutations and so on. And you were like, oh, wow, this method seems to be working. Again, this is physics-based MD simulations, and that's wonderful. It's an example. Let's go now to AI, just to give you another example.
I mean, my question would be based on that example that you chose. Are there no examples on people doing simply empirical experiments to evolutionize an enzyme and they haven't achieved 6,000 improvement on the enzymatic activity?
They do, but it takes a lot of time and money. And that's the reason why nowadays, if you are going to publish about directed evolution, people are looking forward that you are using AI, and you can use basic regression, right? To make it faster and cheaper. If you want to publish a new study without AI in enzyme evolution, the impact will be lower in the journal. People are looking now for novelties. I've been reviewing papers on that in Nature Communications and other journals, and they are now looking into not only how to use AI to train basic models, even with a few data points, even with 50 mutations, you can do machine learning. It's more active learning, right? It's not deep learning where you need like 10,000 data points. You can do active learning and not just one property, but actually 2 properties. So this is where people are moving towards. Can you make the method more efficient? Because ideally, you will be able to predict, like with protein PNN. I mean, why do you need evolution? People don't want to do evolution. They just, Sergio, just make 5 genes. You improve your expression, solubility, stability. I don't care about evolution. I want about something that works at higher temperature, that is better, that you have yield, because that will also be a cost factor. And so where I think is the field moving is, okay, how can you predict KCAT? Well, we can't. I mean, it's very difficult. How can you mutate far away from the active site and control something you don't understand? We don't know how, these loops move, how the substrate moves, enters, goes away. How can you accelerate this transition state stabilization and so on? And we can generate data points. You can use microfluidics. You can use genetic selection. You can sequence, deep sequence with nanopore, with Illumina. You get data points, and you will be able to kind of predict that, but you don't understand it, right? If we want to understand it, this is what I'm so excited, and this is also I wanted to mention this today, is you go and do these MD simulations, but you cannot just do it with these 100,000 atoms. That's very, very expensive. We were using, 200 GPUs, and we just did femtoseconds, just moving a little bit like this. So I think that here's the future. Microsoft has been publishing a lot of stuff on MD simulations of proteins. They are now entering into enzymes. DeepMind is just going to work with enzymes. David Baker is moving to enzymes because we don't understand them. And this complexity I show you, So that means that the breakthroughs, if you're back to your question, what I think the breakthrough will come when you can understand how these things move and how you can predict, a given property or properties. Yeah. Yes, please.
First of all, thank you for your nice talk and the information that you shared with us.
Yes.
In addition to Gabriel's question and complementing, I'm not in that field, but what about the genetic selection?
Mm-hmm.
I mean, if you need to go fast and low cost,
yes,
your design of genetic selection should have an upgrade gigantic in terms of efficiency of those
yes,
genetic selection. And Could you comment something about that?
That's a real question because that's exactly the same with microfluidics. Like you say you can do one experiment with a lot of variants, but you need to put a lot of time and effort to get there in the first place. So this strain we use for the FDH was a knockout of 5 genes that in central metabolism that make NADPH. And you can see here when you have the wild type, I think it's in black. Well, here I don't know what is the color of the wild. I don't think you can see. But then you have single, double, triple, quadruple. When you have mutated those 5 genes and you just add gluconate will this gluconate 6-phosphate will be converted to ribose 5-phosphate by this enzyme in the cell, and it will make NADPH. So 5 mutations were done by an expert relatively fast, like in a couple of months, right? You will always use this strategy to recover the cell. Then you check colony PCR. Then you check that you have no longer those 5 genes in the chromosome. I don't remember how exactly it took for Stefan, the Lidna, to do this many years ago. He's an expert. Let's say half a year, few months, check colony PCR. If it will be just focus time in a company, you can get this into 1 month and a half. Now we have recombination tools that are really efficient to make this in E. coli, for instance. Now, when we use these 5 knockout strains, and they were trying to do selections, they were cheaters. There is always this in evolution. There were cells from E. coli that were surviving without having selection, without selection pressure. And then they sequenced the strain and they found that there were mutations in another enzyme that was having cofactor for NAD to NADP. Just one mutation in the active site was enough to produce NADPH to, overcome that selection, right? That means that they have to do adaptive laboratory evolution. So they cultivated the strain 6 months and they realized, okay, these are the mutations that are coming. So they deleted also those genes. And then they were able to have a strain that works well. I mean, we have been using this strain actually in my group because I was realizing, okay, if you already created, just use it, right? And so now we are trying to generate data with this strain. But it's a good point. It's not just like this, right? It can be very tricky.
Yeah, thank you.
Yeah, you're welcome. Mm-hmm. Yes, sir.
A more philosophical questions like, do you think with the age of the protein design that we are now living, we are able or we will be able to simplify metabolic pathways? Because you were showing us for that like 20 enzymes that you need to select, improve. Yes. Do you think we are in the moment that we can simplify metabolic pathways or even design metabolic pathways specifically for some compounds?
Oh yes, definitely. And actually the best example would be to look into the papers from Tobias Erb, who has been, studying how can you fix CO2 by integrating different enzymes. For example, they published a catch cycle in 2016 in Science. There were 16 enzymes from different organisms that like new to nature, right? I mean, the enzymes were not really existing in nature, but his group repurposed them to be able to fix CO2 to, it was, I think, at some point 4-5 carbon atoms, molecules. And if you think about that, actually, it's a great question because in principle, if you will be able to design enzymes, let's say you have these 20 enzymes, right, a pathway, but if maybe you can do it with 6 that create new chemistries, I think this is where also the future is. And there is also another example by Codexis. I think they engineer 5 enzymes by evolution, and then they cascade pathways, right? I mean, because then you can replace the chemical steps that people will do by enzymes, right? And so they create these cascade pathways to produce some valuable drug, and they are commercializing that actually. But they have to engineer the 5 enzymes by evolution, but in principle, you could do also the Novo protein science. So I guess these people were, if you want to innovate, to work where nobody's working, I think that would be a great example, how to get access to those pathways. And by the way, yes, that could be interesting. For example, I didn't mention, but biotin has this radical enzyme, biotin synthase. People have been trying to work on another path because improving those enzymes is going to be very, very hard. But because of that, of closer. So how can you get actually new chemistry to get to the same biotin molecule? So I know people have been working on that. Yes. Yeah, great question.
Another question? No, thanks, Carlos. Okay.
Yeah, thank you.